Our last module for the week is basic biological concepts. And the objective of this is to give you a refresher and a refresher of prior concepts that you had in biology and an expansion to particular aspects that will be useful as we consider viruses. I will start this week and we'll continue with basic biological concepts in the first part of next week as well. I'll start out by talking about the key molecules, DNA, RNA, and protein. For some of you, hopefully for most of you, this is a review. Then I'll talk about what a virus is and finish up with how we detect and measure viruses and what do we mean by a living virus. So I'll start up with a key concept that underlies really all of modern molecular biology and goes back decades and decades. DNA, deoxyribonucleic acid, is the storehouse of all the genetic information of an organism. In bacteria and higher organisms, DNA is double-stranded. It gets transcribed. You can think of DNA as a master copy of the information. It gets transcribed into RNA, ribonucleic acid. And the ribonucleic acid, specifically the messenger, mRNA, is used as a template to translate the genetic information into the formation of proteins. And the proteins form the backbones of organisms, and many of the proteins are enzymes, which carry out the important metabolic functions of organisms. I'll say a bit more about conversion of RNA back to DNA via reverse transcription a bit later. DNA, deoxyribonucleic acid, has the backbone of carbohydrates, sugar phosphate backbone. It's a double helix in formation, and every rung of that double helix contains either the pair of adenine and thymine or the pair of guanine and cytosine. Those two sets of bases are complementary to each other. For every adenine, there's a thymine. For every guanine, there's a cytosine. Which essentially means that each one of those strands, DNA, if we were to pull the helix apart, contains the same information. So there's redundancy. Looking in a bit more closer molecular detail, if we take the double helix and unzip it, you notice how some of the bases are coming apart at that point, forming a single strand of DNA. We can now take each one of those single strands of DNA and transcribe it into a strand of RNA. The only difference between DNA and RNA is that the thymine in DNA is replaced by uracil. But otherwise, there's a pairing on the RNA. Every adenine on the DNA becomes an adenine on the RNA, and so forth. Once the RNA is transcribed, then the RNA can be read by organisms into a code. Every 3 adjacent bases on an RNA corresponds to an amino acid. And so, for example, GUA on the RNA, guanine, uracil, adenine, corresponds to the amino acid valine, and so forth. And this chain of amino acids may be many hundreds or a couple thousand, forms a protein that's functional. So we go from the master copy of DNA to an intermediate messenger RNA to protein. A virus is the simplest biological form that contains information necessary for its replication. The next several slides I'm excerpting from a lecture on the web by Professor Roncanello at Columbia. Viruses are all around us. Every living thing, whether it's a plant, an animal, an insect, a bird, a fish, or even bacteria, can be infected by viruses. If it's a bacteria, we happen to call it a bacteriophage, P-H-A-G-E. When we eat food, we're regularly eating and breathing billions of virus particles. And in fact, the viruses, when we ingest them, some of them can be carried along as part of our own genetic material. Now, only a small number of viruses that are capable of infecting us that we ingest or inhale or maybe get exposed to on our skin are harmful. Most of them have little or no impact on our health or well-being. They're just being carried along for the ride. And as this slide shows, and we'll talk about it a bit more later, viruses can exist in a variety of shapes, from filamentous forms to compact, almost spherical forms to things like that, which are icosahedral, and so forth. And the particular shape that a virus has is associated with the particular type of virus that we have. So most viruses just pass right through us. A sample was done of supermarkets in DC, and it was found that every 100 square centimeters of leaf from a lettuce or a cabbage contained 10 to the 8th particles of virus that were pathogenic for a particular plant pathogen on the virus. In other words, in this case, this is an example of a virus that actually, from a cabbage's point of view, is beneficial. It helped infect and keep down this particular insect that's pathogen. If we examine viruses in our human waste, the large abundance of them are similar to plant viruses. And in fact, the virus that's called the pepper mild mottle virus that infects peppers, which are a portion of our diet, can occur up to 10 to the ninth per gram of dry fecal material. So it can be extremely abundant. So a virus is an infectious, meaning capable of invading and reproducing in a host. Obligate intracellular parasite. Obligate means that's the only way it can live within cells, consisting of genetic material, either DNA or RNA, often surrounded by a protein coat, sometimes in addition by a lipid, by a fatty membrane as part of the coat of the virus. So now, in addition to the shape of the virus, we have what type of genetic material is in the virus, whether the coat of the virus is protein only, or also has a membrane, often called an envelope, around it. When the virus is outside the host, we call it a virion, the infectious particle itself. Within the infected cell, the nucleic acid can and does hijack the machinery of the cell to produce more copies of itself. Viruses are very small. So this figure illustrates E. coli, which is a common bacterium, magnified 100,000 times. We would have to magnify by 1,000,000 times or more to see a virus. So 1,000th, roughly, the size of a bacteria. Very, very small. In fact, while we can see bacteria under a light microscope and very, very large cells, either animal cells or plant cells, even by the naked eye, viruses can only be imaged by an electron microscope. So quite small. Anywhere from, at the upper limit for very large viruses, 1 micron, all the way down to hundredths of microns or tens to hundreds of nanometers. As the graph on the bottom shows, the figure on the bottom, we have viruses such as polio that are quite small, 30 nanometers, herpes virus, 300, our friend coronavirus is right in the middle, 50 or 60 nanometers. We know a lot of details about viruses. First of all, you can come up with a chemical formula, which is interesting, but perhaps not very useful. We can classify viruses by the type and the sequence of nucleic acid in the virion, by the nature of the protein shell, the capsid, which is the name for the protein shell, whether it's symmetric, asymmetric, what's its shape, does it have particular outcroppings, such as these guys, does it have a lipid membrane or an envelope, and what the size is. And by the way, these spikes that you see on the outer portions of viruses are very often what the viruses use first to attach to the cells that they will infect. And then some of them will actually use those spikes to pierce their way into the interior of the cell and thereby infect it inside. Just like other biological organisms. We can use taxonomy to classify viruses. Kingdom, phylum, class, a number of orders of viruses, many families. This is an illustrative classification for the Ebola virus in the family Filoviridae. Philo meaning filamentous in this case, because Ebola is a filamentous virus, long hair-like structure. Genus, and then finally species. And the Ebola virus that most recently erupted is called the Zaire Ebola virus. Now, one specific class of viruses, which were really only discovered in about the 1970s, are called retroviruses. So we talked about the central dogma. The central dogma says genetic information resides in DNA, which is transcribed to RNA. And the RNA in turn is translated to protein. But retroviruses carry their genetic information in RNA, not in DNA. So the first thing they need to do when they invade a host is convert RNA to DNA by a special enzyme called reverse transcriptase. Let's go back earlier to the slide I had up. This is reverse transcriptase here, going backwards from RNA to DNA. So retroviruses, retro meaning reverse, have to undergo that first step. Coronaviruses are retroviruses. Influenza viruses are retroviruses. A number of important pathogens, as well as non-pathogenic viruses, are also retroviruses. HIV, human immunodeficiency virus, is a retrovirus. Now I want to switch to how we detect and measure viruses. And what do we mean by seeing if they're viable? And in fact, the concept of viability for viruses still remains controversial, because some will assert that viruses cannot be living, since they can't exist outside, they cannot reproduce outside a host. But to my mind, that just makes them an obligate parasite. So there are two basic approaches to detecting and measuring viruses. One is to see if they can be cultured. Can we deliberately infect a susceptible host with viruses and monitor for the infection? A second approach for retroviruses is to directly testing for RNA, or if we were dealing with a non-retrovirus, DNA. So this is a figure from a very recent paper with one of the first culture results for SARS-2 coronavirus. So what you see here are two different plates with animal cells. The brownish background are cells originating from African green monkeys that were grown on a petri dish on a special medium. Then once that lawn of host cells was grown, samples that contained virus were placed on top of it and solidified with agar. And every clear zone that you see represents a location where those African green monkey cells were infected, killed, and the clear area represents the absence of them. So in principle, every single clear dot represents the progeny from one viable virus or perhaps one clump of viruses that went on the plate. And you see if we go from day 2 to day 3, the clear zones get larger and larger. And there are 2 different cell lines here, Vero E6 and Vero CCL81. As is common, there's a difference in performance of different cell lines. Vero E6 shows more visible and more dramatic plaques under these conditions. These assays, as shown, take days to complete. In addition, there's often the question as to whether or not every single virus that's live produces one plaque. We don't necessarily know that. And further work to determine that for every single virus assay needs to be done. In addition, it may very well be, if we're talking about a real sample, there may be other things in the sample that inhibit the infection with the animal cells and inhibit the formation of the plaque. So we need to test for that, perhaps by repeating the assay at different dilutions, or with the use of other material to reduce inhibition. Plaque assays are often considered the gold standard, but they require special facilities, special equipment, and well-trained technicians. In contrast, we can look for the nucleic acid in the case of retroviruses RNA directly by the use of what's called the polymerase chain reaction. Now, for retroviruses, the first thing we need to do is use reverse transcriptase to convert RNA to DNA. We take our sample, we collect it, we purify it, perhaps concentrate it, treat it with reverse transcriptase to produce DNA, and then we put it in this assay. The first step is to unzip the double-stranded DNA that's formed with reverse transcriptase. We use what are called primers, segments of nucleic acid, usually anywhere from 100 to a couple hundred, maybe even to 1,000, that are highly selective for the particular sequence that we want to look at. We want to be sure that we only amplify, which is what this process is called, the target organism that we're focused in on. So you can think of this as a selective photocopy machine, where the selectivity is governed by how those primers are selected and developed. The primers bond to the DNA. We add other nucleotides to the mix to extend the DNA from the primers. So now, from one molecule of DNA, we have 2 molecules of DNA. We unzip each of those again, do the same thing. So we go from 2 to 4. And then if we do it a third cycle, 8, and so on and so forth. So imagine doing this for 20 or 30 cycles. We can amplify this one molecule of DNA by millions of times. And now if we detect how much DNA we've amplified, we can relate that back to the original amount that was present in our sample. The technology we use is called either quantitative PCR, qPCR, or digital PCR. And I don't want to go into the details of those methods, but if anybody is interested, raise it within this discussion, and I can put down some references if you want to dig into that technology. Now, this only tells us if RNA is present, not the viability. This test can be done in hours. And in fact, this has been the primary test that's been used for clinical examination of coronavirus in patients in the current pandemic. The reason why it only tells us if RNA is present and not the viability is we might have enough fragments of RNA in our sample, either in a non-viable or killed virion, or maybe RNA by itself in the absence of a virion, which would not be capable of infecting and reproducing in a host. So theoretically, this is an upper limit for how much RNA is present, and in turn, how many organisms might be there. Selecting the primer, selecting the conditions for this amplification to occur are very important. That will give us selectivity to be sure that we only pick up what we want to pick up and not some other nuisance organism. So that's the first segment of basic biological principles. Next week, I'll finish up with the basic biology, talking more about the properties of the SARS coronavirus 2. I'll talk about prior pandemics, prior outbreaks, and we'll go from there. Remember, go to the discussion if you have questions. I have open office hours on Wednesday, and so those of you that want to chat with me there, feel free, and have a good week.